Today I'll be presenting on DiGeorge syndrome. It is caused by a microdeletion in the region 11.2 of the Q arm of the chromosome 22. That's why it's also called 22q11.2 deletion syndrome. It is considered a primary immunodeficiency caused by abnormal development of the pharyngeal patches during embryonic life, and it affects 1 in 3,000 births, which is more common than cystic fibrosis. And clinical symptoms include immunodeficiency and hypocalcemia due to hypoplasia or underdevelopment of the thymus and parathyroid glands, facial anomalies, cleft palate, delayed growth, poor muscle tone, and cognitive and psychiatric disorders like ADHD and schizophrenia. DiGeorge syndrome is not inherited. The three main genes that affect this disease are TBX1 gene, CRKL, and COMT. TBX1 is responsible for signaling to neural crest cells to promote cell growth and differentiation. CRKL is an adaptor protein coding gene, which is related to thymic, parathyroid, and cardiovascular defects, and COMT gene is responsible for psychiatric conditions such as schizophrenia. Therapies include reconstructive surgeries, transplants, and medications, but there is no cure or cellular treatment for DiGeorge syndrome. The experiments I will be working on are bacterial artificial chromosomes to target a particular gene, which in this case is DGCR2, which encodes for an adhesion receptor, and cells will be collected from a mice model that is homologous to human chromosome 22, and the cells will be put in blastocytes and then into pseudo-pregnant female mice, and the outcome should be that the offspring should have normal development of pharyngeal pouches. And then the adenoviral vector experiment will basically eliminate all symptoms of DiGeorge because the adenoviral vector will contain all the missing genes of DiGeorge, and they will be transfected into blastocytes and then into pseudo-pregnant female mice, and the offspring should have no symptoms of DiGeorge. Thank you.